Hello, hello, good evening. Hi. Yes, yes. Hello, this is me, your biology teacher from Go Score, Alka Rajimon. Welcome back to yet another session of our YouTube live free classes. Yes, I hope there are no technical issues. Is everything good? Okay. Please be active in the comment section. I hope you all are here. I hope there is no visual issues. There is no any audio issues. I hope everything is good. So please give me a thumbs up if everything is okay. And be active in the comment section for the entire class. It's just a 1 hour class, right? Okay, since your board exams are here, I hope you have already made use of all the SQPs and PYQs that we have already discussed. And this is another PYQ session, an SQP session, sorry, SQP session. And today we'll be discussing 2021 question paper. So we have already covered up almost all the SQPs. We have started from 2025, the recent SQP, and today we are here with the 2020 and 2021 SQPs, from which we'll be discussing entirely the biology topics. So I think without wasting much time, we can start, right? Yes, thank you. Okay, so we'll begin. Let's begin the session without wasting our time. So this question paper would be a little different from the remaining question paper because this is not a recent one. As you can see, it's 2021 question paper. So the pattern of the question, the arrangement is all a bit different from the recent question paper. So don't be bothered. All we need is just some questions, right? We need some sample questions. So we'll do the sample questions, we'll solve the questions, and please don't be confused about the question pattern. It's a bit different. Leave it. Okay. So this is your section A. The first question is, veins are thin-walled and justify the statement. Veins are thin-walled, justify the statement. So how are we going to answer this question? This is just a 1 mark question. It will be there in the MCQ part, since I told you this is not exactly MCQ, not one word MCQs, but then you have to write it in sentence because this is an old question paper, but still we need the questions, right? Okay. So veins are thin-walled because the veins are the blood vessels which carry the deoxygenated blood, right? Veins will be, oh, sorry. Okay. Okay. So veins are the one which will be carrying the deoxygenated blood, right? And they will be carrying the deoxygenated blood from the heart, and they'll be bringing back the deoxygenated from the entire body, right? So the blood which is being in the vein won't be under any much pressure when you're comparing it with the blood in the artery. So when you're comparing your veins blood pressure with an artery's blood pressure, the veins blood pressure would be much lower. So if the blood pressure is lower, then the veins being thin-walled is absolutely fine. That is exactly the answer for the question. And they've also said something about valves. Why do veins have the valves? So one thing is they're carrying a low pressured blood, right? They carry a low pressured blood. So vein being thin walled is absolutely fine. But arteries being thin walled, it can break because arteries are always carrying high blood pressure, right? And then why do the veins have valves? Valves are nothing but they are the part where the backflow of blood will be restricted, right? So it is to prevent the backflow of blood, that is the deoxygenated blood and the oxygenated blood, or the blood which is already pumped shouldn't go back, right? So it is to prevent that backflow of prevent backflow. So just to prevent the backflow of blood, that each and every vein in our body has a valve, especially the veins which are carrying blood to and from the heart. Okay, so that's pretty much about it. Moving on with the next question. How is the wall of small intestine adapted for performing the function of absorption of food? And it has an all question. Let it be there. First, we'll solve the first question. So how is the wall of small intestine adapted for performing the function of absorption of food. I hope you all know about this thing. So we all have the entire alimentary system here. This is an exact diagram which is taken from your NCERT reader. And this is your small intestine. And if you're taking a look into the small intestine, you can see that inside this long meters of tubes of vessel, there are these finger-like extensions inside it. There are this finger-like extension inside the small intestine, and these finger-like projections are called the villi, right? These finger-like projections are called the villi, and villi are the ones who are responsible for increasing the surface area of your small intestine. They increase the surface area of your small intestine. That means more villi, more absorption. That's it. More area, more absorption. That's it. So the major function or the major adaptation that small intestine has is the presence of this villi, which are finger-like projection, that will be increasing the surface area of your small intestine. That means increasing the efficiency of digestion. That's it. Then the all question, out of a goat and a tiger, which one will have a long or small intestine? Justify your answer. Again, it's the same pattern of question from the same topic. They're asking about how a goat's sorry, how a goat's small intestine is different from a tiger. That's it. And why they're different. That's it. So as you know, tiger is a carnivorous animal. It will be eating a lot of meat, which is very easy to absorb in the intestinal area, right? We don't need much long small intestinal area or longer absorption time for absorbing meat because it's just protein. It can be easily digested with some proteases, some protein digesting enzymes are enough. But then in the case of goat, as you know, goats are what? They are herbivorous animals, right? So the herbivores in the herbivores, since goats are herbivores, okay, what are herbivores? Herbivores animals or the herbivores, that group itself. So the herbivores are animals who eat only just the plants, right? The grass, the plants would be their entire food. So the plant or the grass family has this content called cellulose in their wall. Plants have cellulose in their wall. The cell walls are made up of cellulose, and cellulose is a compound which needs much bigger digestion time because the bonds between the cellulose is really hard to break. So they need more time to be in there in the small intestinal area to get digested. So that's why the grass eating animals or the herbivores animals have a very long small intestine so that the cellulose get digested properly. So the answer to the question is, out of goat and tiger, which one will have long or small intestine? The goat, because they are the herbivorous animals. That's it. Now next question, explain how ozone, being a deadly poison, can still perform an essential function for our environment. Explain how ozone. So ozone gas, as a gas, it is a deadly poison. If you keep inspiring the O3 gas too much, you will die. It's a poisonous gas. But then the same deadly poisonous gas can be essential for the normal functioning of our environment. How come? How come? So O3 is the gas which is present in the stratospheric region. So what is stratosphere? Stratosphere is a layer just above the troposphere, that is above the earth. So suppose this is the earth. Earth has a single layer of troposphere above it where we can see all the birds flying. That's the region of troposphere. And above that we have the stratosphere. This is the stratospheric region. In that stratosphere there is a zone called the ozonosphere. There is a zone called the ozonosphere. So this ozonosphere, which is a part of the stratosphere, is where this O3 layer is formed. And how is O3 formed? O3 is formed when the oxygen molecule in the stratosphere splits when there is UV radiation and it splits into oxygen atoms. and one of this oxygen atom will react with another oxygen molecule to form an entire O3 molecule. And such plenty of O3 molecules together hand in hand with bonds between them form the ozone layer. So this ozone layer which will be formed in the stratospheric region, this ozone layer which will be there in the stratospheric region will act like a protective blanket from all the unwanted radiations coming from the outer space. So this O3 layer is a protective blanket. That's how the same O3, which is a deadly poison, can still perform an essential function in our environment. That is being a protective blanket from the unwanted radiations. Okay. Then the next question is, give reason why a food chain cannot have more than 4 trophic levels. So why does a food chain doesn't have more than 4 trophic levels? I think we all know the law of 10% law, right? We know the 10% law. What does the 10% law say? So 10% law is an ecological law which states that when energy is moving from one trophic level to a higher trophic level, only 10% of the energy will be transferred. Only 10% of the energy will be transferred to a upper trophic level. Almost 90% of energy will be lost, and that losing is in the form of maybe that animal itself uses that 90% energy, and some loss can happen when it is being transferred. Either ways, only 10% of efficient energy will be moving from a producer to a primary consumer, or a primary consumer to a secondary consumer. Anyways, there is a 10% loss. So by the time energy reaches the tertiary consumer, the amount of energy available would be very zero, or you can say it would be negligible, almost equal to zero. So that won't be enough for another trophic level to be sustained in an environment. So that's the reason why there won't be, it's impossible to maintain more than 4 trophic level. I hope it's clear. It's just the 10% law, the laws of energy. Next question is, state the role of pancreas in digestion of food. State the role of pancreas in digestion of food. I think we all know this answer by heartedly, right? We all know this answer. Yes. Hi Akash, how are you? So the role of pancreas in digestion. So what is pancreas though? Pancreas is this particular organ here, right? So this particular organ, which is almost a leaf-like organ situated just near to your stomach. That is called the pancreas. And the same pancreas, this act like an exocrine gland and an endocrine gland. So it can act like an exocrine as well as an endocrine gland. So, as an endocrine gland, it will produce all the hormones. Okay, it will produce all the hormones, and we know which all hormones, especially the insulin, glucagon, are the ones that pancreas produce. Let it be there. We need not to explain that particular portion here. But then the exocrine part of the pancreas produces a lot of enzyme. It produces the pancreatic juices. So it produces the pancreatic juices, pancreatic enzyme. And this juice has a lot of enzyme. It has the lipases, it has the amylases, it has proteases. So all these enzymes which are produced by the pancreatic system will be taking part in the upcoming food absorption. So the major role, the question is, state the role of pancreas in food digestion. So you can say that the pancreatic juice which will have all the lipases. So lipase is an enzyme which is present in the pancreatic secretion, which will be helping in the breakdown of fat. So that will help in the breakdown of fats. So breakdown of fats and lipids, you can say. Fats and lipids. Then there is another enzyme called the pancreatic amylase. It's called the pancreatic amylase. So pancreatic amylase is an enzyme which is just like the salivary amylase, the same function, but it's present in pancreas, that's all. So this is the one which will help in the breakdown of the starch, you can say. It converts the starch into simple sugar units. So, and then there is one more, lipase, amylase, and then we have the protease. So the protease is another enzyme which is present in the pancreatic secretion that will help in the breakdown of proteins. So the proteins can be converted into the small amino acids. Amino acids is a simpler unit of protein. That's possible with the help of protease. So this is how the pancreas is participating in the digestion process. That's the function of pancreas. That's it. Okay. Yes. Hi. Okay, then we have one assertion and reason kind of question. Let's see, not just one, we have multiple assertion and reason in the same question paper. So let's see the first assertion and reason. Assertion, food chain is responsible for the entry of harmful chemicals. Food chain is responsible for the entry of harmful chemicals in our bodies. That is true, right? It's because of the food chain, it's because of the presence of a food chain that we get all the chemicals which should be present in a water body or in a fish. It is all coming to our body just because we have a food chain in our ecosystem. So yes, that is a true statement. Okay, let me change the color. Okay. So assertion is true. The length and complexity of food chain vary greatly. Yes, even that's fine. Food chains can be very small like grass, grasshopper, frog, snake. That's a food chain. It can be grass, grasshopper, what else? Some insect eating bird, then an eagle, then maybe some, yeah, eagle would be fine. So depending upon the participants in the food chain, the length of a food chain can vary. Even that's also a true statement. But then, is it explaining the assertion? Is the reason good enough to explain? No. In the first assertion, they're talking about the biomagnification process we have studied, and in the reason, they're explaining something else. So you can say assertion and reason are true, but reason is not the explanation of assertion. So it can be B. Okay. Then next assertion and reason. A great number of individuals are present in the lower trophic level. We know that if you take an ecological pyramid, the lower part where all the producers are seen, that's the area where large number of mm, mm, number of participant, what do you call it? The number in an ecological pyramid would be very high in the lower trophic level, and that statement is true. The flow of energy is unidirectional. That's also true. Energy can only move from a lower trophic level to a higher trophic level. That's true. But is it explaining the assertion? No, because they're telling in the assertion, they're talking about the number of things in the lower trophic level and it is high. Why is that number high? Because they get a lot of energy, they have enough and more chemical energy which is being converted from the light energy. So they are available in a very high number. But in the reason, they're not explaining anything about that energy availability there. They're just talking about the flow of energy is unidirectional. That's all. If they have added a sentence like because the lower energy, the lower trophic level has a lot of energy, then it would have been a reason. But still, this one is not a reason. Although the statement, the reason is true, the reason is not a correct explanation for that particular assertion. Then another assertion and reason, a geneticist crossed a pea plant having a violet flower with a pea plant having white flower. So this is something we normally do. A violet flower and a white flower cross. He got all the violet flowers in the first generation. Yes, that's possible. Here, when you're crossing this, all we would get is a capital V small v, which is a violet flower, and all 100% of the F1 generation would be violet. Fine. So the first statement is true. White color gene is not passed on to the next generation. Yeah, even isn't that right? Yes, that is right. The white color gene is not being transferred from this is the white color gene. Small v is the gene for white color. That white color gene is not passed on to the next generation. Here there is only capital V, small v. There is no small v in the F1 generation. And that is the reason why all the plants in the F1 generation are violet. Yes. So assertion and reason are true, and reason is the correct explanation for assertion. So that should be option A. Fine. Moving on. So read the following and answer any 4 questions. So this is a different question pattern. You don't have to take care of the pattern. Just leave the pattern behind. All we need is the question. Let's solve the questions. Okay. All living cells require energy for various activities. This energy is available by the breakdown of simple carbohydrates, either using oxygen or without using oxygen. So they're talking about the topic respiration from our chapter life process. So first question, energy in the case of higher plants and animals is obtained by dash, breathing, tissue respiration, organ respiration, digestion of food. It's none other than tissue respiration, right? That is where the ATP formation is happening inside the cell. Yes. The graph below represents the blood lactic acid concentration of an athlete during a race of 400 meter and shows a peak at point D. So this is the graph. So this is an athlete's respiration graph. Okay, it's showing how much lactic acid is accumulated in his body. So at a point D, there is a high peak too. Fine. Now the question is, lactic acid production has occurred in the athlete while running in the 400-meter race. Which of the following process explains this event? So the process of lactic acid production. What is that phenomenon behind that lactic acid production? I think even without looking into the options, we can answer it, right? Still, there is the option anaerobic respiration. It is during the anaerobic respiration that the lactic acid would be formed inside our body, and it happens only in our muscle cells, and this happens when there is a lack of oxygen. When oxygen is lacking in our muscle cells, when we are taking a huge cardio workout or maybe a marathon, that's when this lack of oxygen can happen, right? Okay, so that's B. Study the graph below that represents the amount of energy supplied with respect to the time while an athlete is running at full speed. So this is X is showing, the X axis shows the time and Y axis shows the energy supplied to that athlete. Okay, so there is two plot. There is a blue colored plot B, which is showing a high rise in the energy in the beginning, and then it falls off. The energy falls off as the time is moving on. And then there is the plot A, which is in red color, that shows a gradual rise in the energy, and the energy is staying constant as the athlete is doing the raising or workout, whatever it is. So choose the correct combination of plots and justification provided in the following table. So we just have to find which is correct combination. So they say plot A is aerobic, plot B is anaerobic. Reason, amount of energy is low and inconsistent in aerobic and high in anaerobic. No, that's completely wrong. It is not inconsistent energy in aerobic. As you can see here, the aerobic always, after that peak of energy formation, it shows a consistency. The plot A shows a consistency. It's maintained. The energy is maintained. But then in plot B, it just falls off. So then aerobic plot A, anaerobic plot B, amount of energy is high and consistent in aerobic and low in anaerobic. Yes, that can be the correct answer, right? So plot A, this plot A should be showing the aerobic respiration, high energy and consistency, while plot B should be showing anaerobic respiration or anaerobic condition, where there is a huge rise in the energy, then it drops. It's not consistent. So it's option B. Fine. Then another question related to the same question, sub-question. The characteristic processes observed in anaerobic respiration are presence of oxygen? No way. It is anaerobic, no oxygen. Release of carbon dioxide? Yes. Release of energy? Yes, release of lactic acid. Yes, if it is lactic acid fermentation, lactic acid will be there. Energy will be released, but the amount of energy produced in an anaerobic respiration would be very less when you're comparing it to the aerobic respiration. That's all. But still, energy is there. So the option is 2, 3, and 4. 2, 3, and 4. Yeah. Option C. That's it. Moving on. Study the table below and select the row that has the incorrect information. So we have to find the incorrect information. All the remaining 3 are correct. So let's see. Aerobic and anaerobic. In aerobic respiration, the location is the cytoplasm, and anaerobic, it's a mitochondria. End product of aerobic, CO2 and H2O. That is fine. Ethanol and CO2, totally fine. Amount of ATP is high in aerobic, fine. Low in anaerobic, fine. Oxygen is needed in aerobic, yes. Not needed in anaerobic, yes. So the wrong statement. Aerobic respiration takes place in the mitochondria. Mitochondria, the powerhouse of your cell. That's where the aerobic respiration happens. And anaerobic respiration is where cytoplasm. It's happening in the cytoplasm of your cell. It's just the vice versa. It's the other way. Aerobic in the mitochondria and anaerobic in the cytoplasm. That's the wrong statement. Okay, now this is section B. Bile juice does not have any digestive enzyme but still plays a significant role in the process of digestion. Justify the statement. It's a 2-mark question. So it can be simply explained by saying what the bile juice is doing. That's it. So the bile juice is produced by the liver and it is stored in your gallbladder. So bile juice is produced by your liver and it is stored in your gallbladder. And the major function of bile juice is to convert the acidic food in the stomach. In our stomach, there is what? HCl. It's acidic condition. So the major function of bile juice is very alkaline in nature. It's alkaline in nature. So the major function of bile juice is to convert this acidic food coming from the stomach into an almost an alkaline medium. So the first function is it creates an alkaline medium so that the pancreatic enzymes can act properly. So the pancreatic enzymes act only in an alkaline medium. So bile juice is the one who converts the acidic food coming from the stomach into an alkaline thing so that the pancreatic enzymes can act on it. So that's the first thing. And the next thing is the bile salt. That's the bile enzymes in which the bile salts are present. They are taking part in the breakdown of large globules of the fat into small globules of fat. So whatever large fat molecules are there, that will be broken down into small pieces. Or you can also call it as emulsification action. Emulsification is the second function. Emulsifying means it's just mixing up like a soap. It just makes the food more fluidy and mucus-like so that it can be easily digested by the lipases in the next step. So one thing is providing alkaline nature. Second thing is emulsifying the fat globules. That's it. Then next question. It's an all question. In birds and mammals, the left and right side of heart are separated. Give reasons. We know that the left and right is there so that the left will carry the oxygenated and right will carry the deoxygenated blood. Separation of deoxygenated and oxygenated blood is necessary so that the animal can maintain its body temperature, so that there is enough energy in the body which won't be lost in the form of heat. So there is no mixing up of oxygenated and deoxygenated. So the answer can be explained in this way. The separation keeps the oxygenated and deoxygenated blood from mixing, allowing a highly efficient supply of oxygen to the body. This is useful in animals that have high energy needs, like the birds and mammals who fly, who run, who are always in a fight or flight mode. They need a lot of energy. So that energy maintenance is possible with the use of that high energy, high body temperature. Okay, so that body temperature maintenance is possible by not mixing up the oxygenated and deoxygenated blood. That's it. Next question. State the events occurring during the process of photosynthesis. Is it essential that these steps take place one after the other immediately? It's a direct question from your text. So what is all the processes happening in photosynthesis? Absorption of sunlight, converting of light energy to chemical energy. It's all there in the text. It's direct question from the NCERT. So this is how they expect you to explain the answer for the 2 marks. So CO2 plus 6 CO2 plus 6 H2O gives C6H12O6 plus 6 O2. So that's the basic equation for photosynthesis in the presence of sunlight and chlorophyll. So you have to explain the steps like this. First step is absorption of light energy by the chlorophyll. That's the first step. Then this light energy will be converted to a chemical energy. That's the second thing happening there. Now, using that chemical energy, splitting of water molecules into hydrogen and oxygen will happen. Water is split. And then finally, reduction of carbon dioxide happens, and that is how the carbohydrate or the C6H12O6 is formed here. That's it. So this is a direct question from the textbook, and there is one more question. Is this step, is it necessary that all these steps should happen one after the other? Is the sequence important? So the steps need not to take place one after the other immediately. Why? Because in desert plants, these steps are not happening in the same sequence. Why? In desert plants, they do not take the carbon dioxide in the daytime. They take it in the night time. So desert plants take the CO2 in the night time when they open the stomata without the sunlight, and they convert the carbon dioxide and they fix it into some form in the night time. So that is what happening first. And then in the morning time, when the desert plants get enough sunlight, they take up the remaining things like splitting of water, release of oxygen. It all happens in the morning. But the carbon dioxide reduction, that fixation of carbon dioxide happens in the night. So that is an example which you can use to explain that all these steps need not to take place one after the other. That's it. So that's a 2-mark question. Then this is your section C. After self-pollination in pea plants, with round yellow seeds. So this is the same dihybrid cross that we have done from our NCERT. It's a direct question again. So let's see. So after the self-pollination in pea plants with round yellow seeds. So round yellow, it's heterozygous round yellow self-pollinating. I hope you remember it from the text, right? So when you're doing this cross, the following types of seeds were obtained. Round yellow, round green, wrinkle yellow, wrinkle green. That is in the ratio of 9:3:3:1. That is the dihybrid cross genotypic ratio, right? Sorry, phenotypic ratio. So that's it. Now the question is, analyze the result and describe the mechanism of inheritance which explains these results. So you just have to explain about the dihybrid cross. What all points do you have to explain there? You have to explain it in such a way that, so somehow when the capital R, capital Y, small I and capital R, capital Y, small y was crossed, you will get the gametes like capital R, capital Y, capital R, small y, small r, capital Y, small r, small y from one parent, and from the other parent also you will get the same kind of gametes, right? They're similar. Capital R, capital Y, capital R, small r, capital Y, small r, small y. And when you finally make the cross between these two parents, you will get these round yellow, round green, wrinkle yellow, wrinkle green in these particular ratio. So there you have to explain that when the dihybrid cross is done and final result is obtained, none of the offsprings are exactly showing the genotype of the parent. They all have their own particular segregation happened. Independent segregation happened in such a way that all of them have received some new allelic combination. So the allelic combination in the F2 generation was not at all present in the parental combination. So the parental genotype is completely different from the offspring's genotypes. That's a major point you have to put in there, and you have to explain the third law of inheritance, that is law of independent assortment. So all these alleles have independently assorted in such a way that none of the alleles are resembling their parents' allele combination. That combination is completely different. That is what you need to explain in precise manner. So this is a model of how you should explain the answer. The ratio is 9 is to 3 is to 3 is to 1, in which parental as well as the new combinations are observed. This indicates that progeny plants have not inherited a single whole gene set from each parent. So, from both of the parents, none of the progenies have obtained a complete same genotypic sets. None of the genes from the parent is same as that in the offspring. But then in offspring, you can also see the round yellows. The parent is round yellow. In offspring also, you can see round yellow. So there is parental combination. Along with that, you can see a whole set of different combinations of the alleles. And that is due to the independent assortment happening. So every germ cell takes one chromosome from the pair of maternal and the paternal chromosome. When two germ cells combine, segregation of one pair of character is independent of the other pair of character. So you have to explain the law of independent assortment there too. So with that, you will get that 3 marks easily. That's it. And there is an O question to that. It is from the sex determination. In humans, there is a 50% probability of the birth of a boy and a 50% probability of a girl will be born. Justify the statement on the basis of mechanism of sex determination in human beings. I think this is a very cliche question, and in all the SQPs that we have solved so far, there was this question, right? So this is a cliche, frequently repeated question, I guess. So make sure that you know how to answer it properly. Again, it's a 3-mark question. So they have asked you to justify the statement on the basis of sex determination. So you can explain it with the help of a diagram, or you can explain it in your own words. Anyways, what you have to do is, in males and females, the parent will have XY condition and XX condition in their sex chromosome, right? Sex chromosome is different in the male and the female. That's the basic concept here. And if these parental gametes are being formed, the gametes can be of two types in male. It can be a X sperm or it can be a Y sperm in the case of male, and in the case of female it can only be an X egg. It's a homogeneous condition. There is no difference in the two sex chromosomes, so they can only produce one type of egg. That's it. And then when the offspring, or when the zygote, you can also say when the zygote is formed, when the zygote is being formed here, the probability is one being XX and the other being XY. So there is 1:1 chance of a XX is to XY combination. That is, a boy or a girl being born to a father and a mother is 50% chance. That is 50% chance there is for a boy and a 50% chance for a girl to be born. That's it. So this is the basic idea behind the question, and you can explain it in your own words. Make sure you depict all these particular points about the sex chromosome, how the sex chromosome is different in male and female, how the gametes would be in males and how the gametes would be in females, and the probability of the zygote. So that's the point you have to explain in that 3-mark question. That's it. Now this is another question. Plastic cups were, oh, sorry. Plastic cups were used to serve tea in trains in early days. These could be returned to the vendors, cleaned and reused. Later, kulhards were used instead of plastic cups. Now, paper cups are used for serving tea. What are the reasons for the shift from the plastic to kulhards and then finally to paper cups? So, it's a kind of direct question, you can say, but then you can also use your common sense to answer the question. Still, uh, it's not a very deep in question where you have to brainstorm anything. You can basically use the common things you know about ecology. That's it. So one thing you have to do is the merits and demerits of plastic, merits and demerits of kulhat. Kulhats are the earthen pots, right? The one made with the soil. That's it. And then there is a paper cup. So just the merits and demerits of all these things gives the answer. So you can answer that particular question by using these points especially. See, the plastic cups have made a big issue of pollution, soil pollution, producing all the unwanted plastic, and its non-biodegradable nature is a major issue for changing plastic cups, right? So the cups raised concern towards hygiene, thus they were replaced by the disposable plastic cups. So normal plastic cups were replaced with the disposable plastic cup. Let it be there. But still, the plastic cups have an issue of this non-biodegradability. It doesn't get degraded in the soil. It stays in there. It creates soil pollution. Pollution is another concern. Thus, they were replaced by the earthen pots or the kulhats. And they are made up of clay. It was okay, but then for making the kulhats, we have to take a lot of fertile soil. We use the top fertile soil of the earth so that we are making the earthen vessels, the clay, the pots. So that became another concern. That's when the kulhats were again replaced with the disposable paper cups. And today we even use the disposable paper cups, which is made from the trees, right? Cutting down of trees is one issue, but still, when you're comparing with the issues that the plastic cups and the kulhats are making, cutting down of tree would be a better option. That's why we have finally adapted to this disposable paper cups. So it can be recycled, it is biodegradable, it is eco-friendly, and it is not causing as much as environmental pollution as the plastic or kulhats would have done. So that's the explanation for that 3-mark question. Fine. Now, this is the last 3-mark question where they're using the chapter life process, excretion especially. Explain where and how urine is produced. So, urine is an excretory product, right? It's a nitrogenous excretory product that will be produced through your kidney, right? The structural and functional unit of kidney, which is a nephron, is the part where the urine would be formed. And there you basically have to explain the 3 steps of urine formation. First one is glomerular filtration, or you can say glomerular ultrafiltration. Glomerular ultrafiltration. That's the first step you have to say. So that will be happening in this part called Bowman's capsule, inside which glomerulus are there. That blood vessels will be taking the first filtration part, that is ultrafiltration. Almost all the amino acids, glucose, all the things would be filtered in that particular region. And then there is reabsorption. So even after this filtration happening, reabsorption will take place through these tubules, especially the PCT, the Henle's loop, the DZT. All those portions will take part in the reabsorption of the wanted things. Whatever needed things would have been there in the urine would have been reabsorbed in those areas. And then finally secretion. So even after reabsorbing the wanted stuff, still there would be some excess water or some salt which should be removed through urine. That will be again given back to the same tubules, which will be ultimately given to the collecting duct, and the collecting duct gives it to the ureter, and ureter gives it to the urinary bladder, and through urethra it will be, the urine will be eliminated out. So this is how the entire process happens. What you have to explain is these 3 steps, and this is a way in which you can explain it for 3 mark. So blood passes through the filtration unit in the kidney called the nephron, passes through the glomerulus in the Bowman's capsule, and that is called the ultrafiltration. 1 point. Filtrate initially has the glucose, amino acid, water, salts, and the nitrogenous waste. So that filtration will filtrate all these things into the urine part. Then in the second stage, reabsorption will happen where water, glucose, amino acid, this all to an extent, a lot of this thing will be reabsorbed. And in the final stage, that is secretion. In that stage, whatever was reabsorbed, again there is a chance for all the excess water, salt, urea to all come back and join the urine formation part. So these are the three steps you have to explain. So one is ultrafiltration, second is reabsorption, and the third is secretion. It's also called tubular secretion. Let it be. And this is the section D, and this is the final section in the particular question paper. I told you the question paper pattern is completely different from the recent, so don't get confused with the questions and all. Yes, Gion, hi. And this is a question. Trace the changes that take place in the flower from gamete formation to the fruit formation. So gamete formation in plants till the fruit formation. That's a 5-mark question. So you have to sit and write that entire story of fruit and flower formation. So you can begin the question with the anther, filament, stigma, style, all those things. So it will be like this. Suppose this is a flower and it will have a stigma, style, and an ovary part, right? Let it be there. So that is the female gynoecium. A stigma, style, and ovary will be the gynoecium part of the flower, which is the female part. And then there will be some filamentous anthers. So there is filament and what is this filament and the combination called? So this filament and the combination is called the stamen, right? So this is the male reproductive part. So you can call this as the stamen, which is the male reproductive part. You can mark stigma, style, and the ovary. That's the female reproductive parts. So let it be there. And from there, what all will be happening? The ovary is the part which is the future fruit, inside which you can see ovules. There would be multiple or maybe single. Depending on the plant, there can be multiple or single ovule inside it. So inside the ovule is where the embryo's development will be taking place. So once a plant is ready for pollination, what happens? One pollen grain would come and fall and on this stigma, right? And that particular movement of a pollen grain to the stigma is what we call as the pollination. So if the pollination was successful, what will happen? This pollen grain will sit on the stigma, it will extend its pollen tube, and it will ultimately reach the ovule part, right? And what is happening inside the ovule part? So if you're taking a zoom in session inside the ovule, so the ovule, once there is enough maturity inside the ovule, you can actually call it as an embryo sac, right? So suppose this is the ovary, this is the ovary part inside which are embryo sac or the ovule. Now I'm showing it as a single ovule, and inside the ovule there would be this cells called the antipodal cells. These are the antipodal cells. Let it be there. There would be two nucleus in the center, and that is called a central cell. And there would be synergid cells in the other side. So this is the synergids. And in between, there will be our egg cell. So this is the condition of an ovule. So here we can see the ovary part inside which an ovule, that's actually the embryo sac, the future embryo sac is there. So this is the central cell of the embryo sac, central cell. These are the synergid cells or synergids. This is the egg or the female gamete. എന്താ ചെയ്യണ്ടേ? നിർത്താം. ചാർജിങ് ആയിരുന്നു, ഇതിപ്പോ ലൂസായി കിടക്കുവായിരുന്നു ഇവിടെ.